Hi, my name is Nuratika Mitiaziz, matrix number 100538.
Hi, my name is Muhammad Haikal bin Muhammad Hanif, and my matrix number is 99729.
Hi, my name is Khairunisa and my matrix number is 99394.
So hello and hi, my name is Marty Jamiling, my matrix number is 99626. So in this video, I'm going to talk about the objective of this experiment and also the reflection part of this experiment. So now we go to the objective part. So the first objective is to measure the amount of reducing sugar in different samples. Second one is to use the DNS method, which produce a color change when sugar are present. And the last objective is to prepare a glucose standard curve for comparison.
Next is materials and apparatus. For materials, what we need is cuvette, test tube, and test tube bag, hot bag, glass beakers, micropipettes, and tips. For apparatus, what we need is DNS reagent, rose-shaped salt, distilled water, and tea for sample.
Add DNS reagent, distilled water, and sugar sample into each label test tube according to the table. Start by preparing diluted sample. Mix 1 ml sugar sample with 9 ml of distilled water in a test tube. Add all reagents. Shake well each test tube. Proceed by placing the test tubes in boiling bath for 5 to 7 minutes. After boiling, set aside to cool to room temperature. Continue by adding 1 ml of rose-shaped salt into each test tube. Transfer 2 ml of each solution into cuvette. Obtain the optical density reading at 575 nanometer using spectrometer. Remember to start with blank.
For this practical, we applied the standard curve from 2A to determine glucose content in different dilution of tea. The absorbance values decreased consistently as the sample were diluted, which matched our expectation. However, test tube 5, which was highly diluted, showed a negative concentration suggesting an error due to pipetting or the limitation of the DNS method at low concentration. Despite this, the result for test tube 1 to 3 were within the expected range and validated the effectiveness of method within its detection limit.
So now we go to the reflection part for this experiment. Firstly, we learn how sample dilution affects measurements and realize the need for careful technique to avoid errors in concentrations calculations. We had deepened our understanding of colorimetric assays. The reaction of DNS is reducing sugars to form colored complex. Although we faced some challenges with dilution and low concentrations readings, we managed to solve them as a tip. Everyone contributed and supported each other to get reliable data. Lastly, I learned how to measure sugar in samples using the DNS method. The color change from the reaction with reducing sugar helped us determine sugar levels using a spectrophotometer. It was a simple and effective method for understanding sugar testing.
For conclusions, the DNS method was effective in measuring reducing sugars in beverages, showing a clear link between sugar concentration and absorbance at 575 nanometer. Most results matched expected values, confirming the method's reliability. However, very low concentrations showed errors, likely due to pipetting or reaction issues. Overall, the method works well within normal concentration.